This is part 2 of an ongoing series. If you want to see part 1, then click the link in the card over here to find the Science of Cells at Work playlist. Hello everybody and welcome to the Science of, where today we're continuing our journey through the anime Cells at Work. Last time we covered red blood cells, platelets and mast cells. Today we're going to start looking at the white blood cells that protect the human body against invading pathogens. There are many groups of these white blood cells, otherwise known as leukocytes, and to start with we'll be taking a look at the granulocytes. Granulocytes are a group of polymorphonuclear white blood cells that are characterized by granules that occur in their cytoplasm. These are the most prominent of the leukocytes present in the blood and are larger than white blood cells, being between 12 and 15 micrometers in diameter. These cells have a very short lifespan, only lasting about 10 days or so. But this isn't an issue, as they're produced continuously from stem cells found in the bone marrow. They leave the bone marrow and enter the bloodstream, circulate around the body for a bit, and then die. These tend to be the body's first line of defense against any invading pathogens, but their abilities are limited. And that leads to the need for a secondary line of defense such as T and B cells, which we'll be covering in the next video. Before we take a look at each granulocyte individually, let's take a look at an ability they all share. Granulocytes are able to defeat invading pathogens and infected cells through a process called phagocytosis. This begins with the binding of an opsonin or complementary antibody to the surface of a granulocyte, which I'll be calling a phagocyte from here on out. Once these antibodies are bound to the cell surface antigen, the cell surface receptors on the phagocyte cluster. This triggers the extension of the cell membrane around the target pathogen, causing the phagocyte to extend around it and eventually engulf the pathogen to form a bubble inside the cell called a phagosome. The pathogen is then digested in the phagosome by a couple of granules called endosomes and lysosomes. These bind to the phagosome to form a phagolysosome, where lysosomal hydrolases are released, which break down various cell components. When we look at this process in the show, we see that it's focused a lot more around defeating the invading pathogens rather than just engulfing them like a Kirby enemy, which is a shame. But I can understand why they decide to show it like that. And on the upside, they do show neutrophils eating parts of the pathogens in other episodes. As the most prominent white blood cell, the neutrophils are the white blood cells we see most throughout Cells at Work. Their design is pretty basic, full white in reference to them being the most prominent white blood cell in the bloodstream, and covered in combat fatigues with a little hat with a little flip up panel at the back, which the show refers to as a receptor. This receptor flips up when the neutrophil is close to an invading pathogen. I think I've mentioned receptors enough over the last couple of videos I've done on cells for you to know that all cells have receptors in some sense. Most of these receptors are on the outside of cells, but some are found on the inside, and many of these act as regulators of mRNA synthesis. Neutrophils express specific receptors called the Fc receptors that are primarily involved in the recognition of immunoglobulin opsonized pathogens. Opsonization is the process of recognizing and targeting an invading pathogen ready for phagocytosis, and there are two main mechanisms that can lead to the labeling of pathogens. One is based on antibodies. The other, which we'll focus on, is called the complementary system. The complementary system is composed of over 30 separate proteins that improve the ability of phagocytotic cells to fight invading organisms. This initiates phagocytosis by opsonizing antigens, producing a specific complementary protein called C3b through one of three pathways. The classical pathway, which is triggered by the formation of an antibody-antigen complex. The alternative pathway, which is triggered by the presence of a lipid-carbohydrate complex. And finally, the lectin pathway, which involves mannose binding lectin released from the liver, binding to carbohydrates and mannose residues from the pathogen cell walls. Once CB3 is produced, it will bind to multiple sites on the surface of the pathogen, and then bind to the Fc receptors on the surface of the neutrophil, attracting the neutrophil to the pathogen and leading to phagocytosis. The most important of these Fc receptors are the low affinity Fc gamma receptors. These receptors have a key role in the immune complex mediated activation of neutrophils. However, this requires multiple receptors, specifically the Fc gamma R2A and Fc gamma R3b receptors on the neutrophil surface. Eosinophils make up between 0 and 6% of your total blood cells. Their main role is acting as a major effector cell inside the immune system and as such they have a role in the host defense against parasitic infectors such as nematodes. We first see the Eosinophil in the 4th episode of Cells at Work, focused on food poisoning. In this episode, we see the Eosinophil take on an invading pathogen, but is unable to defeat it. This is a pretty accurate representation of the ability of the Eosinophils compared to Neutrophils. Whilst they can destroy foreign substances such as parasitic infection, their main role is to promote inflammation. The problem is that compared to Neutrophils, the phagocytotic ability of Eosinophils has been found to be less efficient, so it makes sense that they wouldn't be able to defeat the pathogen. Whilst Eosinophils might look like some kind of monster rancher, this outfit may be in reference to the brick red colouring of Eosinophils when they're stained. As well as this, the pitchfork shaped weapon wielded by Eosinophils is actually a 2-pronged Sasumata, which was traditionally a weapon used by samurai. Consisting of a long pole with a wide U-shaped prongs at the end, it was originally used to retain the arms or torso of an attacker whilst keeping a safe distance. This weapon also might be a reference to the bilobe structure of the Eosinophil's nucleus. Later in the same episode, we see the body get invaded by something that's not a pathogen, but instead a parasitic anisaki, a type of nematode that you get when you eat raw seafood. The next fight illustrates Eosinophils' ability to defeat parasitic infections. In the human body, they're able to do this by the use of antibody or complementary dependent mechanisms. In fact, 4 cytotoxic proteins, maltose binding protein, eosinophil cationic protein, eosinophil derived neurotoxin, and erythropoietin proteins are specific eosinophil proteins that are important in defending against parasitic infections. These are all held within the granules of the cell. Erythropoietin proteins form hypohalous acids that cause the HOSCN tyrosine nitration of eosinophil derived granule proteins, or EDGPs. In episode 4, we're also introduced to the basophil. These are the least common type of white blood cell, making up less than 1% of total white blood cells. The basophils in Cells at Work have a relatively plain design, covered in a raincoat and carrying around an umbrella. Their colour is based around basic stains for basophils, which results in the cells being a dark blue colour. Like other granulocytes, these cells are produced in the bone marrow and have a similar role to mast cells, storing and releasing histamine in response to immunoglobulin antibodies. In fact, the only thing that differentiates mast cells and basophils is the expression of the IL-3R alpha chain on its surface. As well as containing histamine granules, basophils also have granules that contain the chemical heparin. Heparin is a blood-thinning substance that prevents blood clots at the site of infection. The next group of cells we'll be looking at come from monocytes. Monocytes are a type of immune cell that are produced in the bone marrow and then travel around the body through the blood until it differentiates into one of 2 immune cells: macrophages or dendritic cells. For today, we're only going to be covering macrophages and then cover dendritic cells when we look at T cells because they're linked together pretty well. In Cells at Work, macrophages take on the appearance of a 19th century nurse or maid, who often wields hammers, and that fits pretty well with the macrophage's role as the body's violent cleaner. In the human body, macrophages are granulocytes that make up between 2 and 8% of all white blood cells. Unlike granulocytes, these mature in tissues rather than in the bone marrow and can last for weeks or even months in the body, far longer than neutrophils that can only last a few days. These specialised cells are involved in the detection, phagocytosis, and destruction of a variety of pathogens. But that's not all. They're also able to initiate inflammation by releasing molecules known as cytokines that activate other cells. Macrophages are able to detect products of bacteria and other microorganisms using a system of recognition receptors called toll-like receptors, or TLRs. These combine to a variety of pathogenic components such as the flagellin protein found on the flagellum of bacteria. Macrophages also have a key role in adaptive immunity by altering the immune system to microbial invasion. After ingesting a pathogen through phagocytosis, the macrophage will present the pathogen's antigen on its surface. This antigen is attached to an MHC class II molecule. This acts as a signal to other white blood cells that although the macrophage is displaying a foreign pathogenic antigen, that it's not going to do harm to the body. When the pathogen is identified, T and B cells are then activated to attack the infected cells. Oddly enough, in the show they illustrate the monocyte being able to switch between a monocyte and a macrophage. This doesn't happen in the human body owing to the nature of cell specification. A monocyte can turn into a macrophage, but not the other way round. If you enjoyed this video, then don't forget to like, comment and subscribe to the channel. And to combat the ever-changing and frustrating YouTube algorithm, make sure you share the video to help my channel grow. If you have any particular scientific subject or topic that you'd like to see me cover in the future, then please tell me in the comments down below. Or if you'd rather, send me a message directly on Twitter. In the next video, we'll be taking a look at the wide variety of T and B cells present throughout the series and seeing how they compare to their real-world counterparts. But until then, this has been the science of granulocytes and macrophages. I'll see you next time. Thanks for watching. If you're looking for more game-based content, then you can join me over on Togglejam Plays, where every Monday, Wednesday and Saturday I show off a different game. Or if you want to support the channel even further, you could also contribute to my Patreon, where you'll get behind the scenes access to the creation of all videos, as well as being able to vote on what the next Science Of video will be.